Okay, guys, so let's talk about glycolysis. If you look at the word, it's telling you exactly what it's doing. It's taking the molecule glucose and it's breaking it or cutting it, right? So this is the first stage of cellular respiration. It can occur with or without oxygen. This is almost identical in all species, so everything does this. And it has three main phases: energy investment, cleavage, and energy liberation. So we're going to talk about each one of these phases. Now, they believe glycolysis is so important because of the fact it can happen in almost every organism and it's identical, that it probably involved in ancient prokaryotes before there was oxygen for them to get ATP. It makes sense because in the early universe, there was no oxygen present. So essentially, this gives us the idea that this is probably one of the original ways that cells made energy. Now, how are we going to essentially make energy? Is we are going to, or how this works essentially, you have this ADP molecule, and then you have some form where within an enzyme, there is this other thing here that has a phosphate. So the P is a phosphate. And if you see adenosine phosphate, diphosphate, means that it's 2 P's. Here, triphosphate is 3 P's. So essentially what we're doing when we're doing the cycling, we're taking the P that is over here, either with a molecule or just a P by itself, and we're adding it to the ADP, making ATP, and then this guy just leaves. So essentially this is how a brunt of our reactions are going to work that we're using. So the 3 phases, energy investment is basically steps 1 through 3 in glycolysis. And essentially it's so important because we are going to take 2 ATPs to actually make a molecule here. So we're essentially trying to get enough energy in here so the rest of it can happen. The second part is what's known as cleavage, and this is steps 4 through 5, right? And essentially this is the part where we're breaking it into 2 molecules. So think of a meat cleaver, right? And this can give you the idea of cleavage, cleavage is the area where you break 2 things into two beings. And energy liberation means for energy to be released, right? And this is steps 6 through 10. So essentially this is where we're going to make a bunch more ATP, and we're going to also make these 2 NADHs, which if you remember are electron carriers. And what I mean by electron carrier, they literally just take in electrons and bring them to someplace else. So this guy yields total 2 ATP. But just like a job, think about it, investment. If I say that I'm going to invest in my company, or I need you as an investor, essentially what I'm asking you is, I need some money to get this started. Can you give me some funds so that I can get things together so that I essentially can make more money later? So here you are starting. This is your startup cost. Your 2 ATP is your initial investment of monetary funds, if you want to think about it in a business. And this is actually paying off. This is what we're making. So this is the energy investment phase. This is not the full. This is reactions 1 through 2. So on here, you can see essentially that in this first, this is glucose, essentially the circle. And then you take some energy in. So this is energy investment right here. And this is 1 ATP. And if you notice, it goes right here. That's why it's called glucose 6-phosphate. Now, you don't need to know all the terms. I just like to go through it so you can actually see what's happening. So this is the first part where they're taking some of the energy for this glucose. So essentially we can keep it going. By adding this energy, now we've made this have enough energy to keep going. Without it, nothing happens, right? And so then we have this next enzyme here that essentially is going to change this circle a little bit and kind of cut it off. If you notice now, this oxygen is here, then you have this little guy over here. This is fructose 6-phosphate, and this is the sugar. Fructose is the sugar you find in fruit, right? So essentially, so far in this energy investment, you've invested 1 ATP here, right? Now at this point, here is reactions 3 for 4 for this, right? So from here, you have your second energy investment. So now you have enough energy here to keep with this reaction. So from here back is energy investment. But if you notice at this part, what's happening here? We are creating 2 molecules, right? So let's go. We're cutting it in half, right? Do you see that? So basically, the second part here is what's called cleavage. Let me go back one. So this guy right here, from here, oops, darn it, to here, this is part 2, which is called cleavage. And you notice, basically, these molecules can be linear as well, right? So you'll have this really long molecule that essentially is a sugar. And then what we'll do is cut it in half. And if you notice, you have 1, 2, 3 oxygens here, carbons, and 1, 2, 3 carbons here. And if you look, you have 1, 2, 3, 4, 5, and 6. So 1, 2, 3, right here. Essentially, you have this guy, this part here, right? Essentially, you can see that's one side and then the other side. Now, the problem with this is that your body can't use both of these. One of these are bad and one of them are good. Essentially, one of them your body can use and the other one you can't. So your body loves this molecule right here, which is G3P. This is why I call this one the hot stepsister. I think about like Cinderella and all those silly stories, right? Your body will use this. So this molecule right here continues on. This just happens to be the ugly stepsister. And no one wants to go out with the ugly stepsister. So the only way that we can take DHAP, or that dihydroxyacetone phosphate, only way that we can make that successful is turning him or her into the hot stepsister. So you have this enzyme right here that just changes things around a little bit and moves it so that this guy right here, right, this one right here can move forward because we do not like this molecule at all. So that's what's happening in cleavage. Now the last part of this, you go to what's known as energy payoff. So this is where our business is making money. So essentially here, you'll notice that you have this NAD plus and it goes to NADH. We're adding a hydrogen there, so what kind of reaction is that? Think gain of electron or gain in general, right? Which is of a hydrogen. Leo says grr. or oil rig. So reduction is gain, right? So this is a reduction reaction. So essentially this guy gets his 2 little electrons and he's like, woo, I'm out of here. And he's going to move on to go interact with someone else. So essentially this part, we make the electron carriers, right? So electron carrier is here. And then essentially we're going to have a couple reactions here. And we're going to make our 2 ATPs here. Now, if you notice this, all of these reactions happen twice. Because what you're seeing is this goes here. You actually do this. This goes here, but then this guy goes here and goes here too. So because you break this in half, everything you see here, you're going to see 2s. Because this reaction is happening twice, right? So this makes 2, this makes 2. Essentially, if we have 2 here in this one reaction, then you're going to say times 2. So essentially, you're making 4 ATPs total. And so this is the whole reaction scheme here, right? And so essentially, you have here, which is energy investment. We have here, which is cleavage. Then we have here, which is energy liberation. And it's actually showing you here, right, that you have the 4 ATPs being made. Because essentially when we cleave, we're going to have the G3P here, and the DHAP is going to change into G3P and then go twice. So essentially what you have here, and this will be good for you to look over, this happens as a review in the cytoplasm. You use 1 glucose, and essentially you're going to use, or these are the reactants, right? So you use 4 ATP, you use 2 NAD pluses, and you use 2 ATPs. This is at the first with energy investment, right? This is what you use, both of these, in energy liberation. And so the products that you essentially make are a total of 2 ATPs, because remember, these, you have 4 ATPs, but you use minus 2 ATPs, because you use 2 to begin with, so you get a total of 2 ATPs here. You make 2 NADHs. From this process, you're going to make 2 waters, and then the end molecule that's going to go to the next one is this guy named pyruvate. Pyruvate is essentially glucose that's been oxidized and cleaved in half. And because you have that where that big one is cleaved into 2 parts, that's where you get the 2 pyruvates. And if you look at this molecule, or this scheme right here, it shows those pyruvates right there at the end. So these are going to go into the next phase. The next phase, before we talk about that, let's talk about what's so cool. There is something in cancer called the Warburg effect. And this is what's so cool about understanding some of these processes. The Warburg effect is the idea that cancer cells will preferentially use glucose versus not using what's known as oxidative phosphorylation. If you remember, oxidative phosphorylation is the last step that makes over 30 ATPs, right? Or 20 to 30 ATPs at the end, where they use something that only makes roughly 2 ATPs, which sounds crazy to us. But if you think about it, in some ways, it makes a lot of sense. Big tumors can't get as much oxygen in them. So that's why they start proliferating or making a lot of cells really fast, but the cells don't die due to lack of oxygen, where our regular cells would. So this essentially makes them more robust in these low oxygen environments. And how this works is in cancer cells, there are 80% of all cancer lines have an overexpression of the enzymes responsible for glycolysis. So that's how they're able to use glycolysis more than oxidative phosphorylation and not die, because they have so many more of these enzymes. So they're able to make a lot of ATP and thus survive. So essentially, there are diagnostic tools you can use to look at cancer cells. And so what you can do is you can take radioactive glucose, drink it, and then they can do an MRI. And essentially what they can look at is these fluorescent are areas that glucose preferentially will go to. So if it's an area that uses a lot of ATP, like your heart or your brain, something of that nature, then it's not going to be too much of a concern. But if there's an area that does not use high levels of glucose or cellular respiration or ATP, then you can kind of think to yourself, okay, this is a possible tumor. Let's go in now and do a biopsy. Or you can look through and go, okay, this is something that is full-bodied now. Let's make the appropriate decisions we need to make. So it does have a lot of kind of applications in science. The second stage we're going to talk about is a breakdown of pyruvate. So essentially here, we are going to have pyruvate come into the mitochondria. So at this point, everything else happens in the mitochondria. It's going to be broken down by this enzyme called pyruvate dehydrogenase. It's a very, very important enzyme. And then from here, you're going to have 2 molecules of acetyl-CoA go on to the citric acid cycle after this, right? This isn't going to produce any ATP. It is just going to produce this electron carrier known as NADH, and it's going to make acetyl-CoA, which we need to go into the third phase of this. So if you see, this is pretty straightforward. It's an easier reaction. Essentially, you have this pyruvate. And how many pyruvates did we make, or pyruvic acids, did we make during glycolysis? 2. So essentially what's happening here is the 2 pyruvates go through these transmembrane proteins into what's known as the mitochondrial matrix. So this is the matrix. These are the 2 membranes. This is the inner mitochondrial membrane. This is the outer. This is the inner membrane space, which just means the space between 2 membranes, right? And then the solution in the middle, kind of like the cytoplasm, is the mitochondrial matrix. And so this guy shuttles into the matrix, and through pyruvate dehydrogenase, you see this NAD plus is there. You make acetyl-CoA, so this guy essentially is here. And then you notice this gets cut off and leads its carbon dioxide. This comes in and attaches, and then we make this NADH that's going to leave and go to part 4 of this whole sequence. This goes in the bloodstream, and we breathe it out. So this is step 2, which is the breakdown of pyruvate, otherwise known as the intermediate step. Okay. So the very last, so this is a review. Happens in the mitochondrial matrix. Remember, you bring 2 pyruvates, 2 of the NAD pluses, and 2 coenzyme A's into this. It's all 2 because this reaction is happening twice, right? And you make 2 NADHs, which goes on to what we know as oxphos, 2 carbon dioxides, which is a waste molecule, right? That we breathe out. And these go to next step. And remember the NADHs, what kind of carrier is that again? It's an electron carrier. So mitochondria are pretty cool things. And I'm going to mention the mitochondria now a little bit because of the fact we're getting into the last 2 stages. And I'm not going to talk about the last stage. I'm talking about all this other stuff first, because when we get into class, I want to be able to review some of this or give you enough time to look at this again. But essentially, the last part, or step 4 of this, can be a bit of a mind-numbing experience. So I want you guys to be nice and fresh when we talk about this later. But essentially, mitochondria are really cool. They're important in a lot of things. Yes, they're important in cellular metabolism, making ATP. But they're way more than that. They're important for storing calcium, and calcium is important in muscle contractions and nerve impulses, the release of essentially neurotransmitters. They're important in making energy, so they help to kind of warm your body if things are too cold. They help to detoxify the areas around, and they're really important in which is called cell apoptosis, which is programmed cell death, which I think the cell pops, right? And so they actually release a molecule once the cell is compromised, or the ATP is. That's basically a cascade of molecules to tell the cell to commit suicide. Now, not all mitochondria look the same. My research was in heart mitochondria, and this was essentially even telling you, even in the same cell, this is a heart cell, right? Where heart cells have these muscle fibers that go through to help with contraction, and then they have kind of like this membrane on it that's kind of like a cell. This is their sarcoma, is their cell membrane. So even if you look at this, do you see how these are yellow and these are red? It's because even mitochondria in the same cell can look different. The guys that were between the muscle fibers that are for heart contractions look different than the ones that are outside that are essentially used for the ATP for all the other functions in the cell. And so if you look at this, you can see these are the muscle fibers here, and this is outside. And if you look, these are kind of oblong kind of things, and these are very, very round. And even how many enzymes they have for respiration are different. You can see here, this is electron microscope. Remember, we talked about that in lab. And you can see the difference. If you look at here, it seems like all these, and so these invaginations here that you can see, right, that come in, those are what are known as crystals. Out here will be that membrane. In between, like these areas, is the matrix. So can you see how these guys have these really long and they're really flat? And then you look at here, and they're very much moving all over the place, right? And kind of, to me, this looks more like hamburger. So these are the ones that are in between the muscle fibers that are for contractions, and these are the ones on the outside. And they do have different rates of making ATP. These guys are actually a little bit faster, and it makes sense because you need a lot of ATP for muscle contractions. So it's something, as I went through, I thought all mitochondria were created equal, and they really aren't. So this last part is that we're going to talk about in this video is the citric acid cycle. It's also known as the Krebs cycle, or the TCA cycle, or the tricarboxylic acid cycle, right? And they all essentially are the same thing. This is where you're going to have the breakdown of pyruvate, or what's known as acetyl-CoA, that's going to go and make a lot of electron carriers. So here is where the complete oxidation of of glucose happens. And if you remember, oxidation means loss of electrons. So at this point, we're going to totally suck all the electrons and all the energy out of this glucose that we can. The citric acid cycle has 8 steps. Each step is basically done by a really cool enzyme. So just imagine if you were in my majors or class or anything like this, you would end up having to know all the enzymes for everything that we're talking about today. But we're not. But what I want you to do is I want you to look at the citric acid cycle, and we're going to talk a little bit about it. So here's acetyl-CoA and water. So this guy is what's coming from the intermediate step. And it's going to combine with what's called xenoacetate. This is already here, right? This plus this with this enzyme called citrate synthase, and they're going to make citrate. So that's the reaction that's going. So basically, this is kind of like he is on a feeder road and he's coming into the freeway. And all the other cars are there too, right? And so he's combining with all the other cars. So if you look through here, the important things to realize is that you have several different areas that you're making NADH, right? And so this guy, all these are going to go out to the last step, or they're going to be for some other kind of reaction. Then if you look, we have ATP happening here, right? And so this is an energy molecule. And the last thing that's important to realize here is we are making one more electron carrier, which is FADH2. And this guy is going to go with these guys. So essentially, this is making, in this one circle around, you're making 3 NADHs, right? You're making 1 FADH2, and then you are making 1 ATP. This happens twice because you have 2 acetyl-CoAs coming in, right? So basically, all of this you multiply by 2 to give you an idea. Now, there are several places in here, if you like to look, right? You are making carbon dioxides throughout here, and these leave our body. So essentially, that's the citric acid cycle there for you. And so what we're going to do is when the next time we talk, we're going to come in and we are going to essentially talk about oxidative phosphorylation. And this again is just a reiteration of the third stage. And you can see we come in with 2 acetyl-CoA, and these essentially, you have 3 NAD pluses, 1 FAD, and 1 ADP per cycle, right? And you have 2 cycles, remember? And so we make a total of all of this combined because remember, each one you have 2 CO2s, you have 3 NADHs, you have 1 FADH2s, and you have 1 ATP per cycle. So when we come in class or for the next lecture, you are going to focus on what's known as oxidative phosphorylation. I will see you guys later.